So let me begin by introducing Nitin. So Nitin Baliga leads a cross-disciplinary team of scientists to address complex problems relevant to global health, personalized medicine, and the environment. His team uses a systems approach to construct predictive models of cellular and molecular networks within pathogens, cancer cells, and environmental microbes to enable biotechnologies to overcome drug resistance, find new drugs, and predict consequences of climate change. Dr. Baliga is also actively engaged in science education and is passionate about developing and disseminating current science concepts and authentic cross-disciplinary practice to schools. His contributions to high school education have had worldwide impact and have won awards. As a professor at the Institute for Systems Biology, he was one of the founding members and currently serves as the director and senior vice president. Welcome, Nitin, and we're looking forward to your talk.
Thank you, Debanu. I'll share my screen. All right, can you see my screen now?
Yes.
Wonderful. Well, thanks for having me here, and good morning to everyone. So my focus is going to be on the idea that the whole issue of precision medicine, especially when applied in a personalized context, is a systems problem. And there are two dimensions in which this problem exists. One is inter-patient heterogeneity, which I'll mostly focus on today. And there's also intratumoral heterogeneity. And if there's time, I'll cover that as well. Now, the challenge in figuring out which therapies would work on a given patient, precision is not the only thing. We have to also address both of the other issues I mentioned. And as I jump into this, first I have to give a disclosure. I am a professor at the Institute for Systems Biology, where I serve as the SVP and director also. And I'm also a co-founder and a board member of a new company that we started called Signomics. And I'll use this opportunity to just very quickly flash this screen for introducing my colleagues in the company. And I'll just jump right into the presentation. So let me try and focus on why we need a systems approach. The current approach to drug discovery has been primarily focused on a drug-centric approach. So large pharma companies are looking for these unicorn targets that could work on a large segment of the patient population. The assumption being that there's an average cancer patient that is going to respond to a molecule that has activity on cancer cells that is developed without any consideration of the unique features of a patient's characteristics. So the pharma companies go through the process of finding these molecules through preclinical screening and clinical trials, and the whole process then gives them a molecule which they have evidence has activity on certain trials that they run that convinces payers to reimburse for those therapies. And then ultimately they go to the patients. Turns out the whole process from the preclinical stage to FDA approval is a very long process. It takes 15 to 20 years. There's very high failure rates in oncology, over 98%, and it's very expensive. And there's some NASDAQ quotes that I've included here if you want to see what happens when a molecule that has been under development for a long period of time fails a trial in the later stage. And the reason why these therapies don't work is because cancer is multigenic and every patient is unique. And it's been figured out that while there's only less than 2% success in clinical trials, if you know something about the patient's tumor characteristics, such as presence of certain mutations, then the success of clinical trials can go up by more than sixfold. So there's a mad rush to find what are the mutations that predict response to therapy. And there's this assumption that there's a linear map of mutation to drugs. But turns out there's not one mutation that exists in a tumor. In an individual patient's tumor, there's hundreds of mutations. Across cohort of patients, there's thousands of mutations. And less than 10% of the mutations are actionable. And within a patient, these mutations act in complex combinations with nonlinear consequences that are difficult to predict. And this is why when drugs are tried on large cohorts, only a small segment of the patient population responds. And it's not clear exactly which patients these are and what are the characteristics. And to address this, we've developed a new approach which puts patients at the center. And based on the patient's characteristics, we can then identify drugs that are likely to work in that given patient and also targets that are likely to be accessible in terms of finding activities for molecules using those patients' tumor cells. And from that information, we can then get evidence to convince providers and payers to use these therapies in a clinical setting, and ultimately using patient-validated therapies, meaning knowing which therapies work on which patients, you can then develop companion diagnostics, do basket trials, and ultimately get evidence with small numbers of patients to go to FDA approval. So how do we do this? So we've developed a platform which can take large amounts of knowledge that exists in the public domain, and we've also generated a lot of knowledge graphs, such as how genes are regulated across human cells by transcription factors and microRNAs, how drugs interact with different targets in pathways, and how they're associated with mutations, how epigenetic regulation plays a role, etc. We've taken the knowledge base, and we can now use it as a prior set of constraints to then navigate through large multi-omics data sets from public patient cohorts. And for example, like the TCGA. And we have developed a suite of algorithms that allows the mining of this information to generate a causal and mechanistic map that tells you precisely which subset of mutations are causally driving cancer phenotypes and through what networks. Now, these are testable hypotheses here, and I won't go into details of how we do this. There's plenty of papers we published on it, but we can do targeted perturbations of the networks that drive cancer phenotypes to validate this model. And once this model is validated, then we can take an individual patient or a small patient cohort and use that patient cohort to figure out precisely what are the genetic programs that are dysfunctional in patients who respond to therapy versus those who do not. And we can do this on an N of 1 basis. So we'll now be able to stratify patients not based on correlates in mutations or gene expression or epigenetic marks, but we can do it based on causal mechanistic network models. And because we have mechanistic understanding, we can then map drugs into the precise networks that have become dysfunctional in a given patient. So this accelerates the discovery of precision therapies matched to patient characteristics. So you can design basket trials or smart trials. And every time you run this trial, you can learn more about where your model was correct, where it was incorrect, and you can iterate on this and grow the knowledge base. So I'll give you two examples. I'll start with multiple myeloma, and if there's time, I'll go to glioblastoma, where we have applied this approach with great success. And starting in multiple myeloma, very quickly, these are our collaborators from academic and commercial. So multiple myeloma, for those who may not be aware, it's a cancer of the plasma cells. About 500,000 patients are affected by it globally, and about 100,000 people die from multiple myeloma each year. And typically the median age is 62 years. And for a number of different types of multiple myeloma, there's a relatively good prognosis with current advances in therapies. But by and large, there are certain groups of patients who just do not respond to cancer therapy. And currently, the way they are diagnosed is by using chromosomal abnormalities. And I'm just highlighting two of the chromosomal abnormalities here: translocation from chromosome 11 to chromosome 14, which is considered standard risk or low risk, and translocation from chromosome 4 to 14, which is considered high risk. Now, depending on these cytogenetic markers, decisions are made regarding how aggressive the clinician needs to be in terms of patient care. It can go anywhere from very aggressive approaches such as tandem autologous stem cell transplant or selection of therapies that the patient is likely to respond to. And I'll show you later, turns out these cytogenetic markers are not precise. And patients eventually relapse. The relapse is the rule. Almost every patient relapses. The first relapse happens in 3 to 4 years, and then the subsequent relapses happen more frequently. And there's a number of therapies that can be selected to treat relapse patients, but there is no clear approach for matching these therapies to patients. So it turns out some patients may never get the right therapy in the end. And in fact, some therapies can make things worse. So we really need to know which are the patients who are at extremely high risk and why. and what are the networks that are perturbed in these patients, so we can then find appropriate drugs to treat their cancers. Similarly, for drug discovery, we can identify the networks that are uniquely perturbed in these patients or not responding to therapy and prioritize those targets for discovery of new drugs using patient-matched cell lines or preclinical models such as xenograft mice and so forth. So we published a paper on this recently. It's in bioRxiv. It is very close to being accepted in Nature Precision Oncology, which lays out how we've taken over 800 patients and analyzed them through this framework I just described to identify a subset of mutations that are acting through this causal mechanistic flow of information to affect cancer phenotypes. There's altogether 13,000 different causal flows that can be affected, but they can be grouped together into 141 transcriptional programs. And all of this will become clear in the next slide. And based on the program activities, we can then group patients into 25 states and associate them with risk. And there's 5 extremely high risk states. So this is the multiple myeloma model. And what you're looking at on the x-axis here are the patients. There's about 800 patients. And on the y-axis here are all the regulons that we've identified. These are subsets of genes that behave in characteristic manner across patients. And a set of regulons are grouped together into a program. The red indicates the programs are overactive, blue indicates they're underactive, and white means they're neutral. And we've grouped about 100 patients into a similar state because the program activity profiles in those patients look similar. Turns out both programs and states have stratifying power. So depending on whether a program is underactive or overactive, the clinical outcomes can be very distinct. Same with states. Patients belonging to a given state can have good or bad clinical outcome. And here are the 25 states rank ordered by risk. And I just wanted to bring your attention to the 2 cytogenetic markers I had described earlier. Translocation 1114, which is considered to be standard risk. Turns out there are a lot of patients who are indeed standard to low risk, but there are some patients, a significant number of patients, who are actually high risk, and in fact, some are extreme risk. These patients get the wrong care with current standards. Same with translocation 414, considered to be high risk. A lot of those patients are indeed high risk, but a significant number are actually very low risk. But these patients get very aggressive care when they probably do not need that aggressive care. And we can look specifically in the translocation 414 patients as to which are these patients who are low risk. And with one program activity, we can find these patients. And when that program is active, we can then look at a second program and use it to substratify the patients into standard or extreme risk. And these are the survival outcomes. And you can see what we really need to focus on in terms of drug discovery are the patients who fall into this red survival curve here. These are the patients who are not responding to any standard of care therapies, and their networks have information that we can mine to prioritize drug targets. And here's a quick insight into how we could find the drugs and drug targets. So I'm showing you 3 panels here. The first panel are targets for current standard of care therapies for multiple myeloma. And what you're looking at in the panel are the different translocations, del17p, amplification 1q, and so forth. And within each of those cytogenetic abnormalities, patients are subgrouped into different risk states. And we have shaded the cells based on the activity of the targets of the standard of care. And you can see there's some, such as translocation 414, where a number of standard of care therapies are not going to work. And in panels B and C, we're showing drugs that are currently being investigated for multiple myeloma and new targets that we've identified that are likely to work in multiple myeloma patients. And here's a quick example to show that patients who relapse, if you look at the standard of care therapy targets in these patients, the targets are uniformly expressed at baseline and at relapse, right? So there's no information you can gather from the gene expression patterns of these targets. But if you move to the network or program activity, you can begin to explain why these patients relapsed. The targets for these standard of care therapies are all downregulated at relapse, and new targets become active. In fact, we can look across the entire set of programs and identify a number of different programs that become active. And I'm showing you genes in 5 programs. Program 68 I described earlier, it's involved in proliferation of myeloma cells. program 134 is also upregulated across many patients. And when you look, I won't go into details here, this program controls the immune microenvironment of myeloma patients. And I had mentioned one of the therapies that gets activated is for carfilzomib, which is used in relapse patients, but we have to now tailor it to only those patients where this program has become activated. Right? And there's all kinds of targets here that we're now exploring to find molecules that are active in these patients where this program has become active. So the same information can be viewed as a network of mutations to cancer phenotypes to drugs. And what you find here is the network that gets activated at relapse contains about 12 druggable targets. 10 of the 12 targets are in what is called the B-cell maturation antigen network. And just last year, there was an emergency use authorization for a GSK drug that targets the BCMA network. Turns out if you look at any individual patient, not all aspects of the network is active. You can find some subset of the networks. And based on that information, you can then select among the therapies that are likely to work on that particular patient. All of this is complex information, and we are delivering this to clinicians in the form of a report where we can give the patient history using conventional means, such as using Tempus or Caris mutation profiles, and further by calculating risk based on the approach we've developed, and also calculating the therapies that are likely to work on a given patient and giving scores and evidence for each of these recommendations. And here's an application of that to 8 patients from the Seattle Cancer Care Alliance, where by standard cytogenetic markers, these patients were stratified. 7 of them were stratified into a high-risk group, and 1 was stratified into a standard risk. Using our approach, we were able to do much better stratification. 21 we predicted was indeed standard risk, but we predicted 3 of the patients who were considered high risk were actually lower risk patients, and 3 were extremely high risk. And if you look at the predicted and actual clinical outcome, it's almost a perfect correlation. There's one patient that we got incorrect. Turns out this was an older patient who chose not to take the standard of care therapies that were recommended. And maybe this is why the patient did not have good outcomes. And so when we showed this to a clinician, the clinician was extremely excited. He said, look, if I had this information previously, I would not even have had the information of double transplants for this 40-year-old patient I saw on earlier this week. And what that means is this patient could have avoided double transplant. Each transplant costs about up to $300,000, and that comes with a lot of additional risks and complications. So even figuring out which patients don't need therapies has enormous values. In addition, those patients who are extreme risk, for them, we could now recommend better therapies and combinations of therapies that are likely to work on them. Okay, so I have a few minutes. I'll quickly jump to glioblastoma. So we took these 2 cancers. Multiple myeloma, as you know, is a liquid tumor. Glioblastoma is a solid tumor. And this is by far the worst cancer, where you have about 100,000 cases globally. And the current standard of care hasn't changed in over 50 years. The median survival is dismal. It's less than 15 months with fractionated radiation therapy and chemotherapy with temozolomide. And the only stratification marker that is known is the methylation of the promoter of the MGMT gene. If you have the methylation, you're likely to have slightly better response to temozolomide. And a lot of trials have been run. Over 400 trials, only 3 FDA-approved therapies have been approved. To give you one example, 60% of patients have EGFR amplification, but EGFR therapies have failed multiple trials. And this is because having the EGFR mutation alone does not predict response to EGFR inhibitors. There's other mutations that could buffer the effect of EGFR amplification. So we need to have a better understanding of which therapies are going to work on which patients, and then prioritize those therapies based on the characteristics, not just at the level of mutations, but also the program activities. So we published a paper on this. I won't go into details on how we built the model, but here's the map of all the programs that can become dysfunctional across multiple glioblastoma patients, and the drugs that could map at each level of these perturbed networks. And of all the drugs that map, we can then hone in on a subset of drugs that are likely to be effective in GBM patients because they're either in phase 4 clinical trials for any cancer or have been tried in GBM trials, meaning there's evidence that they had worked on some patients, but there wasn't enough signal to select these drugs for clinical use. So here's an example. We took a Tempest profile for one patient, and we figured out that this patient had about 27 different program components that had become dysfunctional, and we identified all the upstream and downstream drivers and consequences of these program units, and figured out all the therapies that are likely to work for patients where these programs have become dysfunctional. And here's all the evidence that we have from our own experimental analysis and literature that these are bona fide causal mechanistic flows for glioblastoma using CRISPR-Cas9 screens, using targeted inhibitors, and all kinds of other analysis. In this, I'll give you one example. In this patient, bevacizumab, which is one of the 3 drugs that have been approved for GBM, we found that this drug could work on the patient, and we found a specific flow of information across patients that link mutations to the activity of VEGF-A containing regulon, which is targeted by bevacizumab. So it's a VEGF inhibitor. Having the ATRX mutation would inhibit the program and downregulate VEGF. So all patients who have that mutation would not respond to bevacizumab. Turns out, based on the exome sequencing of this patient has 3 mutations. None of them are in ATRX, suggesting this patient would have increased activity of the VEGF program, making bevacizumab effective in that particular patient. So in this case, you would recommend this drug. Similarly, we have evidence for all the other drugs, and further we can go to combining the drugs. And of course, for drug discovery, now we have all kinds of targets within these regulons that could be exploited with preclinical and early stage molecules that have activities on genes with the right tissues from this particular patient. So this is how we would deploy this in a clinical setting, where a patient would come see a clinician, they would get a biopsy, biobank it, send it for profiling, and we would then use our signal network models to analyze those data and generate a patient-specific disease network map. And then we would map drugs from current standard of care, drugs from clinical trials, investigational molecules into these patient-specific maps to create 2 recommendations. One is to predict risk. How likely is the patient to progress to adverse outcomes versus desired clinical outcomes? And accordingly, what are the therapies that are going to work on the patient? This can then go through a tumor board to a clinician. We can then prescribe this therapy to the patient, and then we can learn from whether the patient did respond or not to keep growing our knowledge base. Okay, so in the last 2 minutes, I'll quickly cover this last idea that for patients who get therapies based on stratification that the way I described, you will get improved response. But for all cancers, especially glioblastoma, you will eventually have a relapse event, because by targeting interpatient heterogeneity, you are not addressing the second issue, which is intratumoral heterogeneity, where every patient's tumor could have all subtypes of glioblastoma cells, which were initially thought to be unique to specific patients. So we've developed a single cell analysis platform and use that to prioritize therapies for individual patients. And this is in an early stage, again, in a review at Nature Precision Oncology. It's a separate manuscript. It's in bioRxiv as well, if you're interested. So what we did is we took one patient and tracked the patient from primary biopsy all the way through recurrence at autopsy. And in the intermediate stage, we grafted the patient's tumor into mice and treated the mice with fractionated radiation therapy and chemotherapy, did single cell analysis over a time course, and then we did analysis of all the single cells across the continuum of samples that we generated. Here are all the single cells from the primary biopsy and the different patient-derived xenograft mice models, and they're color-coded so you can see where the sample cells come from. So the primary tumor cells are primarily in this cluster up here. And when we do radiation and chemotherapy in the mouse model, these tumor cells then start transitioning into transient states and induced states into a recurrent state. And when we take the recurrent tumor sample and map it into this spectrum of single cells, we find they primarily map into the late stage of this trajectory we've uncovered. And what I'm showing in the bottom here in these heat maps are the program activities that become sequentially activated and deactivated as the cells respond to therapy. And accordingly, we can now identify specific drug targets and programs that are selected against, induced, or transiently activated, right? And accordingly, we can now tailor therapies to an individual patient based on how their tumor is going to respond to standard of care therapies and evolve to escape the therapies and go to a new recurrent stage. And these are all the therapies that we identified would work in either a concurrent setting, in an adjuvant or neoadjuvant setting, or a recurrent setting. Okay. So with that, I will stop and take questions. Thank you.
Wonderful. Thank you again, Nitin, for your presentation. I really like that slide deck where you showed the cost analysis of the bone marrow, where you had 100 to 300,000. And that was a very good slide that I personally took a screenshot of. And if there's any questions from the chair or from the audience, feel free. Oh, there is one question. No, it was more of a comment. Sorry, go ahead, Dr. Dabano.
Hi, Nitin. Great talk. I just have one question since I guess there are no others. I'll present my question. It's a lot of work, fascinating work going on in target validation using systems biology, networks, analytics, and so on. Are there any common themes emerging in the underlying concepts in how they are modeled that are bringing them to somehow converge in terms of being able to predict the same kinds of things?
Well, I mean, there's a number of different ways people are thinking about doing what we're doing, but few are actually looking at the causal mechanistic networks. I mean, there is general consensus that we really need to go beyond just mutation profiling, that mutation profiling alone does not give you all the information you need, that you need to go to the other omes, if you will. But there's not much work, at least that I'm aware of, where people are putting these into these causal mechanistic network models into which they're mapping patients. And to my knowledge, this is not being used across the drug discovery spectrum. So at every stage of drug discovery, I believe there's a need to match the molecules to the tissue characteristics. Even when you're doing high throughput screening with millions of compounds, it would be good to, for example, for multiple myeloma, we have 25 states and substates. If you had panels of tissues that represent each of those states and you ran the screens in a smarter fashion, you're likely to get more hits. So the loss across the different phases of clinical trials are going to be less. Similar with trials. Now, phase 1 trials and phase 2 trials are where a lot of failure happens because you use small numbers of patients that are randomized, and you give them a drug assuming they are all uniform, but they're distinct. And so you may get hit and miss results there. And the numbers of patients are not large enough to get enough signal to go to the next stage, and so on. So,
thanks a lot. I did notice that there were a couple of DNA repair proteins in your list of targets in myeloma and both glioblastoma and myeloma, specifically PARP and POL. So have you been specifically looking at involvement of more DNA damage response genes in your network specifically?
We are, yeah. So the PARP example is a good one. So we first discovered that when we were trying to do help a patient who had relapsed refractory multiple myeloma, and we developed that relapse network, and PARP popped up. And when we spoke to multiple myeloma oncologists, most were not aware. PARP had, to their knowledge, not been used. But then we found out that there was a clinical trial with PARP inhibitors run in multiple myeloma. And they did see enduring response in some patients, but not enough to get clinical validation for the trial. So that was a great example for us to then go back and then ask, what were the characteristics of these patients who did respond to PARP inhibitors, and are these the patients that we are identifying as those with the active programs that we've identified. So, and you're right, there's a number of other such examples that we've identified where therapies, the androgen receptor popped up in glioblastoma. And so there are a lot of surprises for repurposing tests. And I think those are the low-hanging fruit. So as you get evidence from that type of an approach, you can then also use the networks for finding new molecules with the same networks.
Great, thanks a lot.
And there's one more question from Dr. Steve Miller from Sandia Biotech. He's asking Nitin, biopsy to tumoroid and screening. Are you interested?
Biopsy to tumoroid to screening, absolutely, yeah. I think, in fact, that is the pipeline you would need. Yeah, absolutely. I think we should get in touch.
Wonderful. Yeah, he's from Sandia Bio, and we can definitely introduce you to him after the program. He is one of our speakers as well later on. And again, I do want to thank you, Nitin, for your amazing presentation. And Dr. Dabano and the crowd, I think we'll be moving to our next program by Dr. Christoph, which is a biological intelligence approach to precision medicine.